first lecture of module 1, we will be talking about peptide bonds. Now, what these peptide bonds do, their characteristics, their specific geometry, and how they are extremely important in holding the amino acids together, the basic framework of what proteins are. So, what we are going to look at is we are going to look at peptide bond formation, their properties, their overall geometry. And the keywords for this lecture would be peptide bond, the formation, the planarity and the rigidity. So, by the end of this lecture, we should be absolutely clear as to how the peptide bond is formed, how it has a planar configuration conformation, and how it maintains a rigidity in its geometry. This is extremely important in an understanding. So, we have the amino acids. In the amino acid, as you can see on the what we have on the left here, let us try and identify this amino acid, considering that we already know the types of side chains that we have. So, the first thing that we have to identify is the alpha carbon. This is the alpha carbon. So, when we are looking at the alpha carbon, we are going to try and understand what are the specific characteristics of this. So, this is the alpha carbon. To the alpha carbon, we have the NH2 group attached. We also have the COOH attached. What else is there? This therefore is the side chain. Now, if we understand what the side chain is now, we see that there is a CH2, you can see the CH2 here, and there is an aromatic ring. To that is added an oxygen and a hydrogen. So, this means this amino acid is tyrosine. Now, this in an understanding of proteins, we have to know the side chains of the amino acids very well. So, after we know what the amino acids are, we have to know their linkages. Their linkages form what is called the peptide. What is this peptide is what we are going to understand. So, the protein then forms this structure that we see on the right here, that is a measure of the folding of this polypeptide chain, which we will see in subsequent classes. So, the protein itself is a polypeptide chain that occurs in nature and folds into a well defined three-dimensional structure. We will talk about folding and characterization and more about the structure in later classes. When we look at now peptide bond formation, so what do we have? We have the amino acids that are going to be linked to one another. So, we have amino acid 1. How do we characterize the amino acid? We look at the amino acid by understanding that we have the alpha carbon, we have the carboxylic acid group, we have the amine group, we have the R group. So, this we know is characteristic of all amino acids. We have now amino acid 2 come. So, we have the N terminus that is given by the NH2 group, we have the C terminus given by the COOH group, and now we have amino acid 2. Now, they are going to link together, and what are they going to do? They are going to condense together in a process known as condensation and lose the water molecule. So, we have OH on this side and H from the amine N terminus of the second amino acid, and we lose the water. So, in this process, we have the formation of a dipeptide, and this is the peptide bond. Now, this peptide bond has very unique characteristics, unique geometrical constraints, which we will understand in this lecture. This process is known as condensation. We can also have hydrolysis, where we can break this peptide bond into the two individual amino acids that it was comprised of. But in this formation, we have to know that very importantly, it is the N terminus and the C terminus of the peptide that is important here. So, whenever we write the sequence, which we will see in a subsequent class, what do we mean by the sequence? We mean by the order in which the amino acids are linked to each other. So, when we have our first amino acid linked to the second amino acid, we know that the amino or the N terminus is on amino acid number 1 and the C terminus is on the end amino acid. In this case, since we have a dipeptide, it is going to be on amino acid number 2. Now, this is extremely important in understanding how peptide bonds form and how the sequence of the proteins is maintained. So, now that we have the peptide bond formation, we understand the beginning of the protein is known as the N terminus and the end of the protein is known as the C terminus. So, what we have is we understand now that this is our peptide bond, amino terminus, C terminus. The peptide therefore is a short chain of amino acids. What we saw was a dipeptide. We can have similarly a tripeptide. What will happen in the case of a tripeptide? In the case of a tripeptide, the COOH of the dipeptide that we saw in the previous slide, this will lose this water and the hydrogen from the amino terminus of the subsequent amino acid that we have here, which will have the R group, which will have the H group, followed again by the COOH. So, when we have the loss of this water in a condensation reaction, we will have another peptide bond formed. So, when we have 3 amino acids linked together, we are going to have 2 peptide bonds linking them. So, a peptide is going to be a short chain of amino acids. The polypeptide, as we call it, because it is going to be a polymer of amino acids, we will call it a polypeptide. So, in the polypeptide, if we look at just the way we can just look at these, we can then have a very long polypeptide chain that is going to fold into a unique 3-dimensional structure that defines what a protein is. So, if we have these beads that are connected one after the other, symbolizing the amino acid type, that is all the information we actually need. We only need to know what R group we have. So, whether it is R1 or R2 or R3, our identity of the side chain is what we need, because we know that the rest of the amino acids is common for all. So, if I know what R1, R2 and R3 are, I know how they are linked together in the polypeptide chain, because they are linked through peptide bonds. So, if we look at this linkage, we have the link through the peptide bonds that tell us that these different kinds of amino acids form this beaded structure one after the other in a necklace formation that will then we will see how this folds into a unique 3-dimensional structure, giving the protein not only a unique structure but also unique functionality. So, the peptide group now is what we need to study in terms of its chemistry. It is a rigid planar structure which is a consequence of resonance interactions. It has 40% double bond character. Now, how does that happen? And it has restricted rotation above the peptide bond. So, this is what our peptide linkage was. How did we get this? We lost the OH from the COOH, we lost one of the hydrogens from the NH2, and we resulted in the formation of a peptide bond. Now, this lone pair of on the nitrogen can actually go and form a partial double bond, and in that what it's going to do, it's going to result in restricted rotation about this double bond, the partial, as we said, of partial double bond character, which renders this a relatively planar structure. Now, this gives a lot of conformational restraints to the rotation about this bond, which is unique to protein structures. So, the peptide bond characteristics include a partial double bond character, and the planarity that we see is due to the delocalization of the pi electrons over the N, C and O atoms as we saw it, keeping a maximum overlap between the contributing pi orbitals. So, this is what results in the partial double bond character, which is important in its geometry, in its constraints in rotational motion. The 4 atoms therefore that we see lie in a relatively rigid plane that is important, or rather restricts the rotation about this bond that has lost its single bond characteristics to a certain extent due to the double bond character formation. Why is that? Because of the lone pair on this nitrogen. Another important factor involved in peptide bonds is this, we will see as we go along in an understanding of the interactions, is hydrogen bonding possibilities between the C double bond O of the backbone and the NH of the backbone as well. When we look at these hydrogen bond formations, they actually are formed between the different parts of the polypeptide chain. So, if we look at a peptide bond on one part of the polypeptide chain, there can be hydrogen bond interactions between different parts of this chain that assist actually in the folding characteristics, which we will see in a subsequent lecture. And the planarity of the peptide bond and its capacity to form hydrogen bonds follows directly from the electronic structure that we just discussed. Another very important characteristic about the peptide bond is its geometry. What is this geometry? In terms of the rotation about the single bond that we just saw. So, the planarity of the peptide bond, as we saw, has the capacity to form hydrogen bonds. Now, this is where we have restricted rotation, this being our peptide bond. Now, if we look at the orientation, as we would say, of the R group. So, this is the way we have our dipeptide written. How do we know this is a dipeptide, and how do we know the direction? We know that it begins from the N terminus, ends at the C terminus. And what we have here is the formation of a peptide bond, because this is amino acid number 1, this is amino acid number 2. Now, if we look at the orientation with respect to the peptide bond, we see that they are trans to each other, meaning they are on opposite sides of this partially double bonded peptide bond. Now, how does that help us, or what more can we understand from this? So, when we have a trans peptide bond, we usually see that peptide bonds assume this trans conformation in which successive alpha carbon atoms are on opposite sides of the peptide bond. So, here is our alpha carbon of the previous amino acid, here is the alpha carbon of the subsequent amino acid, and we see them on opposite sides of the peptide bond. But since this still has some rotational characteristic possible, or some rotational property possible because of its single and the double bond formation, the partial double bond formation restricts its rotation, but it also remains partially in a single bond form, which will allow rotation. And if that rotation does occur, we will form what is called a cis peptide bond. In the cis peptide bond, what do we see? This is our peptide, the C double bond O. This is the C double bond O. This is the NH. So, there has been rotation about the single bond between the C and the N. What are the C alpha atoms here? These C alpha atoms correspond to two consecutive amino acid residues. And what do we observe here? We observe that they are on the same side of the peptide bond, making this a cis peptide. The important or interesting characteristic here is what we see in proline. In proline, cis trans isomerization is an extremely important factor. This is the side chain of proline. As we have discussed earlier, it is an amino acid that folds back upon itself, where you can see the C alpha here, then the C beta, the C gamma, the C delta, and this is the amino terminus of the proline amino acid. So, now if we look at this, if this is the C alpha of the previous one, what do we see about the C alpha of the next? We see a possible trans formation. But what happens in this case is there can be rotation again about this. And then what do we observe? We observe that the two C alpha are on the same side, giving us a cis conformation, which is more prevalent in proline because of the side chain property being an amino acid. So, this gives us some idea of the trans and the cis conformations. So, when we see the C alpha on the same side, we know it is a cis. When we see the C alpha on opposite sides, we know it is a trans. And this gives us a very important geometric characteristic of our peptide bond. So, what we are now going to do, or what we did see, we saw the peptide bond formation, the relative polarity, the planarity, the rigidity, and its isomerism. What we are now going to try and understand is, if you remember in one of the lectures, we tried to find out the pi of an amino acid. But now we have amino acids linked together to form a peptide bond. Now, in the formation of the peptide bond, the characteristics of the individual amino acids also come into play. So, how can we estimate the pi for a peptide? The first thing that we have to know is what are the ionizable groups present, what are the protons that can be lost during the titration of this peptide, and based on that we know that if we go from a charge of +1 to 0 to -1, this the pKa, if we remember what we did for the amino acid, the pKa values responsible from taking the species, be it an amino acid or a peptide, from +1 to 0 and from 0 to -1, it is the average of these two pKa values. So, what we are going to see now is, so here we have our peptide now. So, in our understanding of the peptide, we are going to look at what are the important characteristics here. If we have the carboxylic acids, we know that they ionize at acidic pH. That means they give up their protons. The amino acids ionize at basic pH, which means they would give up their protons at a relatively alkaline pH range. In addition to that, the characteristic of the environment is also important. So, the carboxylic acids near an amino group have more acidic pH. What does it mean? It means that they would lose their proton easily because they have more protons in the environment due to the amino group present. Similarly, amino groups near a carboxylic acid would have a more acidic pKa than isolated amines. So, if we look at a specific dipeptide now and try to determine the pi of this dipeptide based on the information that we already know, let us see how it can be achieved. So, say we have the ala-lysine dipeptide. That means the ala has the N terminus the lysine has the C terminus. We have 3 ionizable groups. One is going to be for the 2 termini, the N terminus, the C terminus. And the third one is because lysine itself, being a basic amino acid, has an ionizable group as well, an ionizable side chain. So, we have an alpha amino group that is comprised or contributed by the alanine. We have a pK of 9.9 for that. The alpha carboxylate group from lysine, because we know the way we have written the dipeptide means that the lysine forms a C terminus. In addition to that, we have a side chain amino group from lysine and we have a pKa associated with that. So, to do an exercise like this, the best thing is to construct a table with all the possible ionizable groups that are present. So, for this case, we have the alpha amino group contributed by the alanine, we have the carboxylate group contributed by the lysine, and we have the side chain amino group contributed by the lysine. And we want to look at the net charge. Why do we want to look at the net charge? Because when we do our calculations, we have to know at what point we are at a charge of -1, what pKa value is taking me to a charge of 0, and subsequently, sorry, this should be +1, subsequently what pK value is taking me to -1. So, I have to go from a +1 to 0 to -1 to determine the pI of the peptide as we have written it. So, at pH 1, what are we going to expect? We are going to expect that the amino terminus is going to be completely protonated. The lysine side chain is going to be protonated and the carboxylic acid group will have not lost its proton. Because why? Because this is going to lose it at 2.2. So, at a pH of 1, we have a +1 charge for the NH3 plus at the amino terminal, the N terminus. We have the side chain +1 because of the NH3 plus on the lysine side chain, and this has a charge of 0 because it is still COOH. Now, as the titration continues, we will, as soon as we cross 2.2, we will lose the carboxylate proton. The COOH is going to become COO minus. This means that at pH 5, what is going to happen? We are going to have a +1 charge for the amino because we still have not crossed this pKa value. This has now gone from COOH to COO minus. So, we have lost this proton. So, the charge is now -1. The side chain amino group is going to be lost at 10.8. So, we still retain the +1. So, the net charge at pH 1 was +2. The net charge at pH 5 is now +1. So, now let us proceed with the titration and see what happens at pH 7. If we look at the values at pH 7, we see that the alpha amino group still does not lose its proton. The alpha carboxylate group has already lost its proton, so it retains this charge. The side chain amino group has not lost its proton, so it is still +1, and the overall charge is still +1. At pH 10 now, we have lost this proton. So, the charge is now 0. This is -1. This is still +1 because we have not exceeded the pK value of lysine. So, the charge now, the overall charge is 0, rendering this dipeptide neutral at this pH. At pH 12 now, we are going to lose the charge on the side chain amino group. So, the alpha amino has already lost its proton. The alpha carboxylate has lost its proton a long time ago because of it's the carboxylate group. Then we have the side chain amino group now that has lost its proton. So, the charge now is -1, this is +1 and this is 0. So, what are we interested in? We are interested to know which pK has caused this transition, which pK has caused this transition, and based on that we will find out what the pi of the dipeptide is. Now, we realize that because of the presence of the lysine group, this is definitely going to have a pi that is going to be in the alkaline range. So, this exercise can be done for a very varied number of peptides, longer peptides. There are now sites available that will actually calculate the pi for you given the sequence of amino acids. But it is good to know how to do this calculation in determining what the pi of a peptide is going to be. So, the references as we have these specific books and our understanding of the overall peptide bond, where we looked at the characteristics of the peptide bond in terms of its geometry, in terms of its chemical characterization, why it has a partial double bond character, and because of this partial double bond character, it has restricted rotation. We have conformations such as the trans and the cis conformation, and we also learned how we can find out or estimate the pi value of a peptide bond or a polypeptide, and understand how we can estimate the charges on the side chains in an example that we did for alla lysine dipeptide. Thank you.